Hello friends, in this video we are going to discuss about platelets. Okay. In this video we will first look upon the development of platelets, then we will go for morphological characteristic of platelet, then we will move to functions of platelet, then we will move for mechanism behind platelet plug formation, then we will discuss contents of platelets. So these are the topics which we are going to discuss in the platelet. Clear? So first, starting with your development of the platelet. So it occurs in the following sequence. First, there will be pluripotent stem cell, as we have discussed in erythropoiesis. So this pluripotent stem cell will divide and give rise to your committed stem cell. Clear? And this from committed stem cell colony forming unit, gem will arise, and this M is for megakaryocyte. Okay, this is for megakaryocyte, and platelets arise from these megakaryocytes. Megakaryocytes will be fragmented to give 100 of platelets. So this is the developmental sequence: pluripotent stem cell, then giving rise to committed stem cell, then colony forming unit gem, then megakaryocyte, and then platelets. Okay. Now we will move for morphology of the platelets. Here, morphological characteristic of the platelets. So the first point is that platelets are about 2 to 4 micrometer in diameter. Here, 2 to 4 micrometer in diameter. Second point is that they are non-nucleated. They have no nucleus. Here, the third important point about platelet that its shape depend on the state of the platelet. That if platelet is in inactive state, if the platelet is in inactive state, then the shape will be your disc shape. Okay, disc shape. And if platelets become activated, then in active state they will acquire irregular shape and having pseudopodia. Different types of pseudopodia is there. Clear? Now the fourth point is about that the platelets are bits of cell fragments which are arises from megakaryocyte, that is your large precursor megakaryocyte. From these platelets arises by fragmentation of blood, as we have discussed in development. Okay. Now, fifth important point about platelets, that it contain actin and myosin, which are the contractile protein which are present in your muscle. Besides these two contractile protein, it also contain thrombostenin, which helps in the platelet contraction. Okay, at the time of platelet retraction, we will discuss in detail about that in protein. Second, it contains residual of your endoplasmic reticulum and Golgi apparatus. Okay. The third point about that, it contains mitochondria, which will lead to formation of your ATP. Okay, mitochondria is present in platelet, which will synthesize your ATP. Next point, it also contain and synthesize prostaglandins. Okay, it synthesize your prostaglandins. Clear? Which are local hormones that may cause many vascular and other tissue reactions. The last one, it contains very important protein, that is a fibrin stabilizing factor, which is related to blood coagulation. We will discuss detail in blood coagulation video. It contains also growth factor. Okay, that causes vascular endothelial cells, vascular muscle cells, and fibroblasts to multiply and grow. So it contains actin myosin. Besides this, thrombostenin also. It contains residual of endoplasmic reticulum and Golgi apparatus. Contains mitochondria, which is used for ATP synthesis, prostaglandin, fibrin stabilizing factor, and growth factor for the smooth muscle cell and fibroblast growth. Clear? Okay. It also have microtubules. Okay. It also have microtubules, which are made up of your protein tubulin. Microtubules are made up of protein tubulin, and they are present beneath the cell membrane, which provides support to the platelet. Okay. The canaliculi, which is present in the platelet, is projected deep into the cell from the cell surface, facilitating the transport of calcium ion. Okay, transport of calcium ion, because calcium is required during your coagulation. Clear? Receptors are also present on the cell surface, which helps in binding with the van der Waals factors. Okay, we will discuss about receptor in a separate discussion. Now we will move to contents of the platelets. We have discussed morphology, development. Now we are going to discuss about the contents of the platelets. So first we will discuss about the content of alpha granules which are present in the platelets. Okay. So first it contains your platelet factor 4, which is procoagulant, which helps in the coagulation. Procoagulant, platelet factor 4. It also contains fibronectin. Okay, it helps in binding. This fibronectin protein helps in binding to traumocyte and prevent your blood loss. Okay, traumocyte, fibrin protein bind to your traumocyte. Okay. The third is your thrombospondin. It also stabilizes your platelet plug. It helps in stabilization of platelet plug, thrombospondin. And the fourth one is your platelet derived growth factor, which promotes fibrosis and wound healing. Clear? So these are the contents of alpha granules. Beside alpha granule, platelet also have dense bodies. Okay, it also have dense bodies which contains your adenine nucleotides plus serotonin. Clear? It also contains lysosomes, which contains different acid hydrolase enzyme. Okay, it contains acid hydrolase enzymes. It also contains microperoxisomes. Platelets also have microperoxisomes, which contain your enzyme catalase. Here, so these are the main contents of the platelets. Now, coming to the receptors which are present on the platelets. So platelets have two important receptors. The first one is your GP1B and the second one is your glycoprotein 2B3A. So these are the two basic type of receptor which are present on the platelet. Glycoprotein 1B, glycoprotein 2B3A. Clear? So first coming to the discussion of glycoprotein 1B. So this receptor of the platelets bind with one bilirubin factor, which is attached to your subendothelial collagen. Okay, which is attached to the collagen. These receptors help in platelet adhesion to traumocyte, because at the traumocyte collagen is exposed. Okay, and because of the exposed collagen, the platelets come and bind there and prevent blood loss. Okay, so this is also known as platelet adhesion. On the other hand, this glycoprotein 2B3A receptor, okay, on the other hand, this receptor bind with similar receptor on the other platelets, okay, and help in your platelet aggregation. So this receptor bind on the same receptor on the other platelet and help in platelet aggregation at the place of trauma. Clear? Now one clinical is also there. If there is defect in your group glycoprotein 2B3A receptor, okay, then it causes a bleeding disorder that is known as Glanzmann thrombosthenia. Okay, Glanzmann thrombosthenia if there is defect in this receptor. Clear? So these are the contents and the receptor. Now coming to the functions of the platelet. Clear? The first function is which helps in hemostasis. We will discuss in detail about hemostasis. It also secretes platelet factors which is needed for your protein such as fibrin stabilizing factor or fibronectin, thrombospondin. These are the factors which are released from platelets, which are the contents of platelet which helps in blood coagulation. Here it also contains your platelet derived growth factor which helps in the growth of your vascular endothelium, vascular smooth muscle. We have discussed about that. Platelets have also phagocytic properties. Okay, they are also phagocytic in nature sometimes. Platelets secrete your serotonin and one more compound, that is your thromboxane A2. Okay, platelets secrete your serotonin and thromboxane A2 which have vasoconstriction effect, which is vasoconstrictor effect. Okay, so these are the main functions of your platelets. Now coming to the last part, that is your platelet plug formation mechanism. Clear? So the main steps are as follows. First, there will be platelet adhesion, then there will be platelet activation, then there will be platelet release, then there will be platelet aggregation, then there will be formation of platelet plug. So they are the basic steps which are followed in platelet plug formation. Clear? Now we'll discuss in detail. First, there is injury to vessel wall which exposes your endothelial collagen. Injury to vessel wall will expose endothelial collagen. Clear? Now platelets get attached to the endothelial collagen by one Willebrand factor which acts like a bridge between them. Okay, so this step is known as platelet adhesion step. The platelet come and bind to exposed collagen as we have discussed in receptors part. The third will be after the platelet adhesion, the platelets become activated. Okay, and these activated platelets will give out your different pseudopodia, and at the same time after activation, discharging of the contents will also occur. Platelets will discharge its content. Clear? And the discharge contents include your calcium, ADP, clear, thromboxane A2, which is vasoconstrictor, thromboxane A2 is a vasoconstrictor, clear? Okay, and the sequence of events leading to your thromboxane formation. We will discuss also about thromboxane, how thromboxane is going to form. We are going to discuss about that. Clear? So your first, that platelet activating factor. Okay, the platelet secrete your platelet activating factor. This platelet activating factor activate your phospholipase C, which will convert phosphatidyl inositol into your DAG plus IP3. Okay, and this DAG activate your phospholipase A2, and this phospholipase A2 will convert your phosphatidyl choline. This is phosphatidyl inositol. This is a phosphatidyl choline. So this phospholipase A2 will convert this phosphatidyl choline into arachidonic acid, and cyclooxygenase enzyme convert arachidonic acid into your PGS2, which is a cyclic endoperoxide. Clear? Which is a cyclic endoperoxide. Now this PGS2 will give rise to three compounds. The first one is prostacyclin. Clear? The second one is PGD2, and the third one is your thromboxane A2. Clear? By the enzyme thromboxane synthase. Here the enzyme is working thromboxane synthase. Here the enzyme is working your prostacyclin synthase. Clear? Now the discharge content, that is ADP and thromboxane A2, both causes stickiness of your platelets, okay, which help in your platelet aggregation. Clear? And then the final step is the platelet plug seals up all the small rents or injury in the vessel wall. But for bigger bleeding site, platelet plug itself is inadequate, and we require clotting mechanism there. Okay, so this is all about your platelet. So thank you for watching. If you like the video, please subscribe my channel and share with your friends.